Good morning, children. Today in my sixth video, I will be taking about clotting of blood or coagulation of blood. Clotting of blood or coagulation of blood is of great importance for the survival of animals. Whenever there is any injury in the blood vessel or cut in the skin, the bleeding takes place. But after a short period of time, the bleeding stops because the blood has the power to clot. So thereby, this clot acts as a plug, thereby sealing the ruptured blood vessel. It stops bleeding and prevents loss of main body fluid. A decrease in capacity of the blood to clot leads to profuse and fatal hemorrhage, that is blood loss, even if the injury is slight. That is, if the clotting power fails in an individual, then the person suffers from a problem called hemorrhage, where there will be large amount of loss of blood at a slight injury. The mechanism of blood clotting, this involves a series of chemical reactions taking place in sequence one after the other quickly. Various events that takes place during clotting of blood we can study in 3 steps. In the first step, we'll study that at the site of injury, whenever any part is injured, at that site, the blood platelets and the injured tissue cells will get disintegrated. They will break down and they will release a chemical substance called thrombokinase or thromboplastin, recently called as Stewart factor or X factor. In the second step, this thromboplastin or thrombokinase will now act as an enzyme and in the presence of calcium ion will convert an inactive form of protein present in the blood plasma to its active form. Inactive form of protein present in the blood plasma called prothrombin into its active form, thrombin. Now, this second step takes place in the presence of an element calcium ion and other factors present in plasma. So here the inactive prothrombin will be converted into thrombin by thromboplastin. Third step, the thrombin formed in the second step will now be responsible to convert a soluble protein present in the blood plasma, which is dissolved in blood plasma. This thrombin will change it into, that protein is called fibrinogen. This thrombin will change that fibrinogen, soluble fibrinogen, into insoluble fibrin. These fibrins are insoluble forms, very fine threads they will change into. And this will then, on polymerization, these threads will form a network of threads which will entangle or will trap the blood cells and will form a clot or a thrombus at that point. The clot is also called thrombus. The thread-like network will contract and squeeze with the blood vessels in it, and the liquid part of the blood plasma will squeeze out. When the clot will form, the network will squeeze, will contract, the liquid will flow down from that clot, and thus we will find that liquid part is called as serum. So that is the third step. Now I can show you this in the form of this whole process by a schematic representation. the blood platelets and the injured tissues, when they both get disintegrated, they release thrombokinase. This thrombokinase, in the presence of calcium ions, will convert inactive prothrombin into active thrombin. This thrombin, again in the presence of calcium ion, will convert the soluble protein fibrinogen into insoluble form fibrin. These fibrin, that is network of threads, on polymerization, this fibrin formed on polymerization will change into fibers. These fibers will form a network and will entangle or trap the blood cells and form a clot. The clot is also called as thrombus, Thrombus. This is the schematic representation of the blood clotting process, and these are the 3 steps that I have explained to you. Now, what are anticoagulants? The substances which prevent clotting of blood are called anticoagulants. In human blood, there is a substance called heparin, which does not let the blood to get thickened or to form clots. Examples of anticoagulant are sodium oxalate or sodium citrate are also used as anticoagulants. Then there is an annelid leech whose salivary gland also contain an anticoagulant, hirudin, which helps the leech to suck the blood from the host body by keeping the blood thin. Now, the whole process of blood clotting will occur in the presence of vitamin K. That is, the role of vitamin K is very important in blood clotting, because it is this vitamin which is going to stimulate the liver cells to produce prothrombin and fibrinogen. And these are the factors which will play an important role in the process of clotting. Now, why does the blood not clot within the blood vessels? Because the blood contains an anticoagulant called heparin, I explained you, and this heparin will not let the formation of thrombokinase within the blood vessels. So once the thrombokinase is not formed, the process of clotting will not take place, because this is a compound which initiates the process of, or which starts the process of blood clotting. Now, what is blood blisters? That is clotting which takes place in the blood vessels under the skin. The skin is not being cut, but the blood vessels is ruptured under the skin and the clot is formed under. So bleeding takes place inside and the clot is formed. That is called as blood blister. So blood blisters are formed without the blood coming in contact with air. These may be formed due to the injuries like sharp pinch when the skin does not break. What is defibrinated blood? That is the blood from which the fibrin has been removed. Once the fibrin is formed, we will remove that fibrin, and the liquid part of the blood plasma with the blood cells is called as defibrinated blood. What is coagulated blood? That is, if we'll take some blood in the test tube and the clot can be seen formed in the usual way, the serum will squeeze out from the clot and gets collected on the surface, and that is called as coagulated blood. I can show you the figure of this. See, we have taken the blood in this test tube. Then we'll find these fibrin, these threads, they are separated, and the clot, forming a clot here, and the serum, that is the liquid part of the blood plasma without fibrinogen, is called serum. It gets separated. This is called as defibrinated blood, or this is called as coagulated blood. If the blood platelets fall to a very low than the normal number, coagulation occurs very slowly, and this may lead to hemorrhage. So number of blood platelets should be maintained. Normal number should be maintained. If the number falls below normal, then the person, clotting power fails and the person suffers from hemorrhage. So example we have seen in the case of dengue fever, where the blood counts of an patient falls below normal. Tomorrow I will be taking blood transfusion and blood groups. Thank you.